Okay, think about this for a second. What if our absolute most powerful medicines weren't actually built by chemists mixing liquids in a lab, but were instead grown? Grown right inside the microscopic machinery of a living cell. I know, it sounds a bit like science fiction, right? But this is the reality of modern medicine. Today, we're taking a journey into these microscopic factories that are quite literally keeping millions of people alive.
Welcome to this explainer. Today, we're breaking down biopharmaceutical production, tracing the incredible journey from genetic engineering all the way to therapeutic delivery. If you've ever wanted to understand exactly how the medicines of the future are being made right now, well, you're in exactly the right place.
Here's our roadmap for today. We'll start with the biopharmaceutical revolution, then look at engineering microscopic factories, scaling up and purifying, real-world medical impact, and finally, we'll wrap up with challenges and future innovations.
All right, Part 1, the Biopharmaceutical Revolution.
So here's the first major shift we really need to wrap our heads around. Biopharmaceuticals are medicinal products produced using biological sources. We're no longer just, mixing synthetic chemicals in a beaker and hoping for a good reaction. Instead, we're literally harnessing the power of life itself, living organisms, cells, complex biological systems to manufacture cutting-edge treatments.
To put that into perspective, on one side of modern pharmacology, you have traditional chemical synthesis. Think of your everyday aspirin. It's a small, fairly simple molecule manufactured through basic chemical reactions. But on the other side, you have biological sources. These are biopharmaceuticals, like synthetic human insulin or complex monoclonal antibodies. These are massive, highly complex molecules that only a living, breathing cell can actually assemble properly.
Now, why does this massive shift actually matter to us? Well, because these biologically sourced treatments give us absolutely unprecedented specificity. They allow us to treat incredibly complex diseases, offering therapies that target the exact root cause of an illness, which dramatically improves patient outcomes. It's literally the key to personalized medicine, finally giving us the tools to treat genetic disorders and cancers that, not too long ago, were considered completely incurable.
Moving right along to Part 2, engineering microscopic factories.
Now that we know what these biopharmaceuticals are, let's zoom way in and see how we actually build them. The production pipeline starts with recombinant DNA technology, which is basically cut-and-paste genetics. First up is gene identification and cloning, where we isolate the exact DNA blueprint responsible for the therapeutic protein we want. Second, we construct an expression vector. Think of this as the delivery vehicle, complete with promoters and regulatory sequences that carry our blueprint. And third, we select the host cell, the living factory that's actually going to read these instructions and build the medicine.
But when we choose that host cell, we usually have to make a bit of a trade-off. Bacterial cells like E. coli are super fast and incredibly cheap to grow. But if your medicine requires really complex protein folding, like a highly sophisticated antibody, bacteria just kind of can't pull it off. For that, you need mammalian cells. And yeah, that often means using Chinese hamster ovary or CHO cells. I know, it sounds bizarre, but these cells are truly the unsung heroes of modern medicine because they can handle the complex post-translational modifications that these advanced therapies absolutely demand.
Okay, Part 3, scaling up and purifying. So, with our microscopic factories engineered and ready to go, it's time to scale things up. Steps 4 through 6 take us straight from the petri dish to the manufacturing floor. In cell culture and fermentation, we grow these engineered cells in massive bioreactors, tightly controlling the temperature, pH, and oxygen levels. During protein expression, the cells do all the heavy lifting, synthesizing our therapeutic protein. And finally, we hit downstream processing, which, honestly, is arguably the most critical and complex part of the entire manufacturing cycle. Downstream processing is all about separating our life-saving medicine from the rest of the cellular soup. We literally have to harvest the cells, sometimes break them apart, and run the material through intense filtration and chromatography. Think of chromatography like a high-tech molecular sorting machine. This step is absolutely vital because it ensures the final therapeutic product is completely pure and entirely safe to put into a human body. The final 2 steps of our 8-part journey are quality control and then formulation and packaging. Once the drug is purified, we formulate it into its final dosage form. That could be an injectable solution, a vaccine, or even a lyophilized, freeze-dried powder that's ready for global distribution. But hold on, before anything gets packaged, it must pass rigorous quality control. Because we're dealing with biological products manufactured by living cells, strict regulatory standards demand these exact tests. We must absolutely guarantee purity,
potency, sterility, and long-term stability. Remember, we are injecting these products directly into patients. A single contaminated batch is a massive public health risk, so the safety evaluation here is totally uncompromising.
Let's move to part 4, real-world medical impact.
All right, so we've successfully engineered the cells and manufactured the drugs. But how are these actually applied in the real world? Just look at the sheer variety of therapies this technology enables. We've got recombinant proteins like human insulin managing diabetes for millions of people. We have monoclonal antibodies actively hunting down specific cancer cells. We have the exact biotechnology behind mRNA and viral vector vaccines. And we even have gene therapy products using CRISPR-Cas9 to go in and correct genetic defects right at the DNA level. These categories represent the absolute front line of modern medicine.
And the impact here isn't just about treating a single patient in a hospital bed. These microscopic factories actually power global public health through massive disease prevention and vaccine programs. They're also the fundamental engines driving modern drug discovery and advanced molecular biology research across the globe. If you take away one overarching benefit of this entire field, it's precision. By targeting the exact mechanism of a disease directly, say an antibody that only binds to a specific protein on a tumor cell, biopharmaceuticals drastically reduce the nasty side effects usually associated with traditional, broad-spectrum drugs. It's literally the difference between using a targeted laser beam and a sledgehammer.
And finally, Part 5, Challenges and Future Innovations.
But, as you can probably guess, building medicine with living cells is definitely not easy. Despite the incredible medical advantages we just went over, we have to acknowledge the real friction here. These biological drugs are exceptionally expensive to make. The manufacturing processes are wildly complex. Plus, biological proteins are super fragile. They often require unbroken cold chain storage and transportation just to prevent stability issues. Making them is really only half the battle. Getting them safely to the patient? That's just as hard.
Navigating all these production and distribution challenges requires strict compliance with global health organizations like the WHO, the FDA, and the EMA. They set the uncompromising standards that ensure these complex, living therapies remain completely safe and effective for patients worldwide, regardless of where they happen to be manufactured.
Despite the hurdles, the future here is incredibly bright. Looking ahead, innovations like artificial intelligence-assisted drug development are rapidly accelerating how we design these molecules. Meanwhile, advanced bioreactor technologies promise to lower those steep manufacturing costs and dramatically increase the efficiency of our cellular factories. We're moving really quickly toward an era of truly personalized biologic therapies and next-generation vaccines.
Which leaves us with this final thought. We've seen how we can turn cells into tiny factories to produce targeted, life-saving therapies. But as artificial intelligence and advanced gene editing continue to combine and accelerate, will the next generation of biopharmaceuticals soon overcome today's cost barriers, democratize these life-saving cures, and finally cure the incurable for everyone? Thank you so much for joining me on this explainer. Keep asking questions, and I'll see you next time.